Plant growth and development. Introduction. All cells of a plant are descendant of the zygote. Development of a mature plant from a zygote follows a precise and highly ordered succession of events. Development is the sum of two processes, growth and differentiation. During this process, a complex body organization is formed that produces roots, leaves, branches, flowers, fruits, seeds, and eventually they die. The first step in the process of plant growth is seed germination. The seed germinates when favorable conditions for growth exist in the environment. In absence of favorable conditions, they go into a period of suspended growth or rest and resume metabolic activities on return of favorable conditions and growth takes place. Growth. Growth is irreversible, permanent increase in size of an organ or its part or even an individual cell. It is one of the most fundamental and conspicuous characteristics of a living being. Growth is accompanied by a metabolic process and occurs at the expense of energy. Plants retain the capacity of unlimited growth throughout their life due to presence of meristem at certain locations in their body. This form of growth, wherein new cells are always being added to the plant body by the activity of meristem, is called open form growth, indeterminate. Root apical meristem and shoot apical meristems are responsible for primary growth, that is, elongation along their axis. In dicotyledonous plants and gymnosperms, the lateral meristems like vascular cambium and core cambium, which appear later in life and cause increase in girth of organs, called secondary growth. Growth is measurable. At cellular level, it is principally a consequence of increase in amount of protoplasm. It is measured by a variety of parameters like increase in fresh weight, dry weight, length, area, volume, and cell number. Phases of growth. The period of growth is generally divided into three phases: meristematic, elongation, and maturation. The constantly dividing cells at root apex and shoot apex represents meristematic phase of growth. Cells proximal to the tip represents phase of elongation. More proximal to the phase of elongation is phase of maturation. Growth rates. Increased growth per unit time is termed as growth rate. It can be arithmetic or geometrical. Arithmetic growth. Following mitotic cell division, only one daughter cell continues to divide while the other differentiates and matures, so a linear curve is obtained. Example: root elongating at a constant rate, mathematically expressed as LT is equal to L0 plus RT. LT is the length time at time t, L0 is the length at time 0, R is the growth rate or elongation per unit time. Geometrical growth. In most systems, initial growth is slow, lag phase. It increases rapidly thereafter at an exponential rate, log or exponential phase, as both progeny cells of mitotic cell division retain ability to divide and continue to do so. However, with limited nutrient supply, growth slows down, leading to a stationary phase, giving a typical sigmoid S-curve. A sigmoid curve is a characteristic of living organism growing in a natural environment. It is typical for all cells, tissues, and organs of a plant. The exponential growth can be expressed as W1 is equal to W0 into e raised to RT. W1 is the final size, weight, height, number, etc. W0 is the initial size at the beginning of period. R is the growth rate, T is the time of growth, E is the base of natural logarithms. Here R is the relative growth rate and measure of ability of plant to produce a new material called efficiency index. Quantitative comparisons between growth of living system can be made by measurement and comparison of total growth per unit time called absolute growth rate, the growth of a given system per unit time expressed on a common basis. Example: per unit initial parameter is called relative growth rate. Absolute and relative growth rates. Conditions for growth. Water for cell enlargement, that is extension growth by turgidity. Water also provides medium for enzymatic activities. Oxygen for aerobic respiration to get metabolic energy. Macro and micronutrients for synthesis of protoplasm. Temperature, optimum range for base growth. Light and gravity also affect certain phases or stages of growth. Differentiation. The cells derived from root apical and shoot apical meristems and cambium differentiate and mature to perform specific functions. This act leading to maturation is termed as differentiation. Example tertiary element. De-differentiation. Living differentiated cells that have lost the capacity to divide can regain capacity of division under certain conditions. This phenomenon is de-differentiation. Example, formation of interfascicular and core cambium from parenchyma cells. Redifferentiation. De-differentiated meristems are able to divide and produce cells that once again lost capacity to divide but mature to perform specific functions, that is, get redifferentiated. Example, secondary xylem, secondary cortex, cork, etc. Development. Development includes all changes that an organism goes through during its life cycle, from germination of seed to senescence. Cell division, meristematic cell, plasmatic growth, differentiation, expansion, elongation, maturation, mature cell, senescence, death. Sequence of developmental process in a plant cell. Plasticity. Plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability is called plasticity. Heterophyly in cotton, coriander, and larkspur leaves of juvenile plant are different in shape from those in mature plants. Environmental heterophyly in buttercup is also an example of plasticity, which shows difference in shape of leaves produced in air and water. Plant growth regulators. Broadly divided into two groups based on their functions in a living plant body. Involved in growth promoting activities like cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. First one, auxin, indole compounds, IAA. Gibberellins, GA3 terpenes, cytokinin, adenine derivatives, N6, furfuryl amino purine, kinetin. Involved in growth inhibiting activities like response to wounds and stresses of biotic and abiotic origin, dormancy and abscission. Example, abscisic acid and derivatives of carotenoids. The gaseous plant growth regulator ethylene could fit either of the groups, but it is largely an inhibitor of growth activities. Plant growth regulators, Auxins. Charles Darwin and his son Francis Darwin studied phototropism in canary grass. F.W. Wendt isolated auxin from tips of coleoptiles of oat seedlings, first isolated from human urine, produced by growing apices of stems and roots. IAA and IBA isolated from plants. NAA24D are synthetic. Physiological effects. Initiate rooting in stem cuttings, promote flowering in pineapples, help prevent fruit and leaf drop at early stages but promote abscission of older mature leaves and fruits. Apical dominance, induce parthenocarpy in tomatoes. 2,4-D kill dicot weeds used to prepare weed-free lawns. Auxin controls silent differentiation and helps in cell division. Gibberellins. Vacane foolish seedling disease in rice was caused by fungal pathogen G. fujikuri. Kurosawa helped understand it. Later, the chemical gibberellic acid was identified. GA3 discovered first and remains intensively studied form. All gibberellic acids are acidic. Increased length of grape stalk causes fruit like apples to elongate and improve shape. They delay senescence, used to speed up malting process in brewing industry. Increases length of stem and yield by 20 tons per hectare in sugarcane. Spraying juvenile conifers with gibberellic acids hastens maturity period. Promotes bolting in beet, cabbages and many plants with a rosette habit. Cytokinin. Skoog and Miller crystallized cytokinesis, promoting active substance, and termed it kinetin, a modified form of adenine from autoclaved herring sperm DNA. Kinetin does not occur naturally in plants. Zeatin, the naturally occurring cytokinin, was isolated from corn kernels and coconut milk. Synthesized in regions of rapid cell divisions like root apices, developing shoot buds, young fruits, etc. Helps produce new leaves, chloroplast in leaves, lateral shoot growth and adventitious shoot formation. Overcomes apical dominance. Promotes nutrient mobilization, helps delay senescence. Helps delay leaf senescence. Ethylene. Cousins help to identify ethylene, synthesized in large amounts by tissues undergoing senescence and ripening fruits. Horizontal growth of seedlings, swelling of axis, and apical hook formation in dicot seedlings is influenced by ethylene. Promotes senescence and abscission in leaves and flowers. Effective in fruit ripening by increasing rate of respiration called climatic. Breaks seed and bud dormancy, initiates germination in peanut seeds, sprouting of potato tubers. Promotes rapid internode or petiole elongation in deep water rice plants. Promotes root growth and root hair formation. Initiates flowering and helps in synchronizing fruit set in pineapples. Induces flowering in mango. Ethiphone is a source of ethylene. It hastens fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits. Thinning of cotton, cherry, walnut. Promotes female flowers in cucumbers, increasing yield. Abscisic acid regulates abscission and dormancy. A general plant inhibitor than inhibitor of plant metabolism. Inhibits seed germination, stimulates closure of stomata. Plays important role in seed development, maturation and dormancy. By inducing dormancy, abscisic acid helps seeds to withstand desiccation and other factors unfavorable for growth. In most situations, abscisic acid acts as an antagonistic to gibberellic acid. Photoperiodism. Some plants require a periodic exposure to light to induce flowering. Such plants are able to measure duration of exposure of light. Long day plants require light period exceeding well-defined critical period. Short day plants require light less than critical period. Day neutral plants. No such correlation between exposure of light, duration and induction of flowering response. Flowering in certain plants depends on combination of light and dark exposures and are also relative durations. This response of plants of periods of day or night is termed photoperiodism. The site of perception of light or dark duration are the leaves. A hypothesized hormonal substance is responsible for flowering. Seed dormancy, controlled by endogenous factors, that is, conditions within the seed itself. Impermeable and hard seed coat. Presence of chemical inhibitors, abscisic acid, phenolic acids, para-ascorbic acid, and immature embryos causes seed dormancy. Man-made measures like mechanical aberrations, using knives, sandpaper or vigorous shaking can break dormancy. In mature microbial actions, passage through digestive tract of animals can break dormancy. Chilling condition, use of gibberellic acid and nitrates can remove effect of inhibitory substances. Light and temperature can also break dormancy. Development in plants can be under intrinsic and extrinsic control. Intrinsic can be intracellular, genetic or intercellular (PGR). In plant growth and even differentiation is also open, as cells and tissues of same meristem have different structure at maturity. PGRs can be having complementary or antagonistic role, which can be individualistic or synergistic. Vernalization. Vernalization is either qualitative or quantitative exposure to low temperature for flowering in some plants. It prevents precocious reproductive development late in the growing season and enables the plant to have sufficient time to reach maturity. Wheat, barley, rye have winter and spring varieties. Subjecting biennials like sugar beet, cabbages, carrots to cold treatment stimulates the subsequent photoperiodic flowering response.